For anyone working in drug discovery, medicinal chemistry, safety pharmacology or clinical development, hERG is more than an ion channel. It can become a program-level decision point. A molecule may look promising on efficacy and still face a serious cardiac safety problem if it blocks hERG and disrupts repolarisation. In this video, we will connect the channel structure and unusual gating to QT prolongation, arrhythmia risk, and the reason hERG assessment matters throughout drug development. Let's start with the channel itself. hERG stands for Human Ether-a-Go-Go Related Gene. The name comes from an observation in mutant fruit flies. A defect in a particular gene caused their legs to shake or twitch when exposed to ether anesthesia, almost like dancing. It reminded scientists of go-go dancers that entertained in 1960s clubs and bars. Its systematic gene name is KCNH2, and it encodes the Kv11.1 alpha subunit. 4 alpha subunits assemble into a tetramer around a central potassium conducting pore, while the beta subunit MERP1, encoded by KCNE2, can modulate channel behavior. Each alpha subunit has 6 membrane-spanning segments. The positively charged S4 segment senses membrane voltage. At rest, when the cardiac cell membrane potential sits at a negative level, the activation gate is closed. Depolarisation moves the voltage sensor and allows the gate to open, creating a path through the selectivity filter for potassium ions. The important twist is inactivation. This cartoon represents the fully formed channel. When closed, the S6 helices cross near their intracellular ends, forming the activation gate and preventing potassium ions from entering the pore from the cytoplasmic side. The selectivity filter at the top of the channel allows only potassium ions to pass and is inaccessible when the activation gate is closed. The channel opens when the S6 helices of the activation gate move outward, forming a continuous path from the cytoplasm through the selectivity filter, allowing potassium ions to exit the cell. However, the channel may remain structurally open while the selectivity filter collapses and stops ion flow. This rapid filter-based inactivation is central to hERG's unusual behaviour. It also helps explain why drug binding cannot be understood from a static channel picture alone. Accessibility and affinity can change with channel state. That link between structure, state and function is what makes hERG so important to both electrophysiology and compound safety. hERG channels carry IKR, the rapid delayed rectifier potassium current in cardiac cells. A voltage clamp experiment makes their behaviour easier to see. The membrane begins near rest at -80 millivolts. It is stepped to +40 millivolts near the peak of an action potential and then repolarises to -50 millivolts. At rest, the channels are closed and the recorded current is 0. Depolarisation starts activation and the current increases. But hERG channels inactivate faster than they fully open. The current during the depolarised step is therefore smaller than you might expect because many channels are already in a non-conducting inactivated state. The distinctive event happens on repolarisation. hERG channels recover rapidly from inactivation and briefly return to the open state before closing. As channels recover from inactivation faster than they close, a large outward potassium current is created precisely as the membrane needs to repolarise. For drug developers, the practical point is that hERG liability depends not just on whether a compound binds, but also on the voltage and channel state under which that interaction is measured. Now place that timing into a ventricular action potential. Following the upstroke, hERG channels activate slowly and inactivate rapidly, so only a small fraction conduct. As repolarisation begins, they recover from inactivation and briefly reopen before deactivating. The resulting outward potassium current helps terminate the action potential and return the cell towards its resting potential. If hERG function is reduced by mutation or drug block, the outward current during repolarisation is reduced. As a result, repolarisation is delayed and the action potential is prolonged. On the ECG, that can appear as QT interval prolongation. Delayed repolarisation also increases the opportunity for early after depolarisations and ventricular arrhythmias such as torsade de pointes. This is the drug development consequence behind the physiology. Blocking a single channel can shift a compound's benefit-risk profile, trigger additional investigation, and potentially limit a program's path forward. hERG also matters in the sinoatrial and atrioventricular nodes. These cells generate or relay rhythmic activity and rely heavily on IKR to complete repolarisation. As in ventricular muscle, hERG channels activate during depolarisation but quickly inactivate and contribute a small current. They recover from inactivation during repolarisation and supply the outward current that helps reset the membrane before the next cycle. When hERG function is impaired, the outward current driving repolarisation is reduced. Consequently, repolarisation slows. Pacemaking can become unstable and the conditions for supraventricular arrhythmias or re-entry may be facilitated. So the safety story is broader than one ventricular waveform. hERG contributes to electrical stability across several parts of the cardiac conduction system. Why do so many chemically unrelated compounds interact with hERG? The channel contains a spacious central cavity below the selectivity filter, with several aromatic and polar binding sites. That combination can accommodate a wide range of small molecules. Many blockers enter from the cytoplasmic side when the channel is open, and they physically obstruct potassium flow through the channel. Drugs can also enter the cavity from the cytoplasmic side when the channel is inactivated. Binding is often state dependent because the cavity becomes more accessible in open or inactivated conformations. Highly lipophilic or amphiphilic compounds may also partition into the membrane and reach the cavity laterally. This druggability creates both opportunity and risk. Intentional hERG block can contribute to class 3 antiarrhythmic action, as with dofetilide or sotalol. But the same permissive binding environment allows unintended block by compounds developed for entirely different targets. That is why hERG testing is critical in drug development. It helps teams recognise a cardiac safety liability early enough to guide chemistry, experimental design, and development decisions. 5 points matter most. First, KCNH2 encodes Kv11.1, which carries the IKR current needed for timely cardiac repolarisation. Second, hERG's slow activation, rapid inactivation, and slow deactivation create its distinctive timing. Third, the current becomes especially important during the final phase of the action potential. Fourth, reduced function or drug block can prolong repolarisation and increase arrhythmia risk. Fifth, the channel's accommodating cavity makes it both therapeutically druggable and unusually vulnerable to unintended interactions. For drug developers, the bottom line is simple. Understand the biology, measure the liability in the right context, and treat hERG as an early design consideration rather than a late surprise.